the genetic evidence that changed everything. Introduction, the question that broke the story. For centuries, the story seemed settled. Historians traced migrations through texts. Theologians preserved lineage through scripture. Anthropologists mapped cultures through language and ritual. And together, they built a coherent narrative about one of the most studied peoples in human history, the Jews. A people scattered across continents. A people bound by religion, memory, and law. A people whose origins were assumed to be understood. But what if that certainty was incomplete? What if the story historians told, the story preserved in sacred texts, and the story inferred from culture, was only part of the truth? What if the real evidence had been waiting somewhere else entirely? Hidden not in archives, not in ruins, but inside the human body itself. Because when geneticists finally turned their tools toward this question, they didn't just confirm old ideas. They broke them. This discovery didn't just add new data. One researcher would later say, it forced us to redraw maps, revise textbooks, and abandon comfortable theories. This was not a refinement of history. It was a reset. The new detectives. The protagonists of this story are not kings, prophets, or conquerors. They are scientists. Geneticists armed not with scrolls or swords, but with DNA sequencers, statistical models, and fragments of bone pulled from ancient graves. For decades, genetics had been used to study disease, ancestry, and evolution in broad strokes. But in the late 20th and early 21st centuries, something changed. Technology advanced. Sequencing became cheaper, faster, more precise. Entire genomes could now be compared across populations. And suddenly, questions that had once been philosophical or theological became empirical. Where did these people come from? How closely related are they really? And how much of identity survives exile, conversion, and time? The Jewish diaspora became a perfect test case. A global population, scattered across Europe, North Africa, the Middle East, Africa, and Asia, separated for centuries, exposed to countless surrounding cultures. If genetics could trace a common origin here, anywhere, it could do it anywhere. Introducing the heroes of the investigation. But this story isn't about the Jews as a single group. It's about many groups, each shaped by geography, history, and survival. Ashkenazi Jews, whose ancestors lived in Central and Eastern Europe. Sephardic Jews, tied to medieval Spain and later North Africa. Mizrahi Jews, whose roots stretch across the Middle East. Beta Israel, the Jewish communities of Ethiopia. And smaller, often overlooked groups in India and China. On the surface, they look different. They speak different languages. They carry different customs. Some historians argued they were barely related at all, linked only by religion, not blood. Others insisted on a shared ancient origin. For generations, this debate went unresolved, until genetics entered the room. Ancient ancestors reawakened. One of the most unsettling developments came not from living populations, but from the dead. Advances in ancient DNA extraction allowed scientists to sequence genetic material from skeletons thousands of years old, bones from the Levant, burials from the Bronze and Iron Ages, remains that lived and died long before modern identities existed. These ancient individuals could not speak, but their DNA could. And when their genetic signatures were compared to modern populations, something extraordinary emerged. Patterns appeared. Threads connected ancient Levantine individuals to modern Jewish populations across continents and centuries. Not perfectly, not uniformly, but unmistakably. For the first time, biblical regions like ancient Israel and Judah were no longer just historical or theological concepts. They became genetic reference points. The central conflict, history versus biology. Here is where the tension begins. Traditional historical models suggested that exile and dispersion would fragment a population genetically over time. Conversion, intermarriage, and assimilation should dilute any ancient signature beyond recognition. Yet early genetic results suggested something else. Despite their dispersion, Jewish populations shared a measurable genetic core. Not a single Jewish gene. That idea is a myth, but a mosaic of shared ancestry. A pattern consistent, with descent from a population rooted in the Levant roughly 2,500 to 3,000 years ago. This alone was enough to unsettle decades of debate. But the real disruption hadn't even begun yet, because when scientists looked closer, when they separated inheritance into paternal and maternal lines, the story fractured, setting up the breakthrough. Human DNA carries two powerful historical records: the Y chromosome, passed almost unchanged from father to son, and mitochondrial DNA, inherited exclusively from the mother. These two lines act like parallel timelines, and when geneticists analyzed them separately, they uncovered a contradiction no one had expected. The paternal line pointed firmly toward the Middle East. The maternal line often did not, especially among Ashkenazi Jews. Suddenly, the clean narratives collapsed. One lineage told a story of continuity. The other told a story of mixing, migration, and adaptation. Two truths occupying the same population, and neither could be ignored. This was the moment when genetic evidence stopped supporting history and started rewriting it. Where the story goes next. This investigation would expose how exile reshaped identity without erasing it, how men and women followed different genetic paths, how language mirrored DNA, and how isolation produced both tragedy and scientific insight. Most importantly, it would force a redefinition of what ancestry even means, not as purity, not as myth, but as survival. In the next block, we follow the evidence deeper, into the chromosomes themselves, where the most disruptive discovery waits. Two genetic timelines, fathers, mothers, and a broken assumption. When geneticists began dissecting Jewish ancestry, they didn't look at DNA as a single story. They split in two, because human inheritance follows two radically different paths, each preserving a different kind of memory. One is carried by fathers, the other by mothers, and together, they would dismantle one of the most comfortable assumptions in historical research. The paternal line, a signal from the Levant. The Y chromosome is passed from father to son, with very little change. Over thousands of years, it accumulates slow, predictable mutations, markers that allow scientists to trace deep ancestral origins. When researchers analyzed the Y chromosomes of Jewish men from across the world, Ashkenazi, Sephardic, Mizrahi, the results were striking. Despite centuries of separation, many shared common paternal markers. These markers clustered not in Europe, not in North Africa, but in the Eastern Mediterranean, the Levant, modern Israel, Jordan, Lebanon. Genetically, Jewish paternal lines aligned closely with ancient Near Eastern populations, groups historically associated with Israelites, Canaanites, and neighboring Semitic peoples. This was not a vague resemblance. Statistically, it was robust. For historians, this was uncomfortable, but manageable. It reinforced a long-held idea that Jewish men descended from a core population rooted in the ancient Near East, dispersed through conquest, exile, and migration. The Y chromosome seemed to validate history, but validation was only half the story. The maternal line, where the narrative cracked. Mitochondrial DNA tells a different kind of story. Passed exclusively from mother to child, mtDNA traces maternal ancestry with extraordinary clarity. Unlike the Y chromosome, it reflects the lives of women, often invisible in written history. When scientists examined mitochondrial DNA among Jewish populations, they expected a parallel result. They didn't get one. Among Ashkenazi Jews especially, a startling pattern emerged. The majority of maternal lineages did not trace back to the Middle East. They traced back to Europe, Italy, Greece, the broader Mediterranean basin. In some studies, over 40% of Ashkenazi mitochondrial DNA could be linked to just a handful of European women who lived roughly 1,000 to 2,000 years ago. This was not a marginal anomaly. It was a foundational contradiction. If Jewish ancestry was purely Near Eastern, how could the maternal line be overwhelmingly European? The old models had no answer. The collision of two truths. At first, some researchers suspected errors, contamination, sample bias, statistical artifacts. But the pattern persisted. Across datasets, across laboratories, across continents. The conclusion became unavoidable. Jewish ancestry did not follow a single, unified path. It followed two. one carried by men migrating outward from the Levant, the other formed by women absorbed along the way. This was not a story of isolation. It was a story of contact, a population that never traveled alone. This discovery forced scientists to confront a reality historians had often downplayed. Jewish communities did not develop in genetic isolation. They lived among others. They traded. They negotiated. They survived under empires that rose and fell. Genetic comparisons showed shared ancestry between Jewish populations and ancient neighbors, Canaanites, Phoenicians, Arameans, and later Arab groups. Not identical, but overlapping. The Levant was not a genetic vacuum. It was a crossroads. And as Jewish communities spread, through Assyrian conquest, Babylonian exile, Roman dispersion, they carried their paternal lines outward while integrating local maternal lines inward. Wars, exile, and the movement of genes. History often tells exile as a clean rupture. Genetics tells a Messier story. Assyrian deportations in the 8th century BC, Babylonian captivity, Roman destruction of Jerusalem and the Second Temple. Each event scattered populations. But scattering did not mean erasure. Men moved. Communities reformed. Local women entered these emerging groups through marriage, conversion, survival. Over generations, genes recorded what texts rarely did. That identity adapted. Why this shattered old models? Before genetics, many scholars argued for one of two extremes. Either Jewish populations were genetically homogeneous, unchanged by time and place, or they were largely converts, sharing culture but not ancestry. The DNA rejected both. It showed continuity without purity, change without disappearance, a genetic mosaic. This forced historians to abandon binary thinking. Ancestry was no longer about either-or. It was about layers, setting the stage for the Ashkenazi puzzle. Nowhere was this complexity more visible than among Ashkenazi Jews, a population that expanded rapidly in medieval Europe, one that would later experience catastrophic loss and astonishing recovery. Their genetic structure bore the marks of this history. A small number of founders, limited early gene flow, and consequences that would echo into modern medicine. But before science could understand disease, it had to understand origin, and the Ashkenazi case would become the most revealing of all. In the next block, we follow this population deeper into language, isolation, and the genetic paradox known as the founder effect. The Ashkenazi paradox, isolation, language, and the founder effect. If genetic ancestry were a quiet background process, the Ashkenazi Jewish population would have remained invisible. Instead, it became one of the most studied and most revealing genetic cases in modern science. Not because it was ancient, not because it was large, but because it was small, and because that smallness left a measurable imprint, a population built from few. Genetic data suggests that the Ashkenazi population experienced a dramatic bottleneck roughly 600 to 800 years ago, a period when a relatively small number of individuals, perhaps a few hundred, formed the core of what would later become millions. Wars, persecution, forced migrations across medieval Europe. These pressures repeatedly reduced population size. When conditions stabilized, the population expanded rapidly, but from a narrow genetic base. This is what geneticists call the founder effect. When a new population grows from a limited number of ancestors, certain genetic traits, neutral, harmful, or beneficial, can become unusually common. In the Ashkenazi case, the effect was profound. Men from the East, women from the West. By this point, the pattern was clear. Paternal lineages traced back to the Near East. Maternal lineages clustered in Southern and Central Europe. The most plausible explanation was no longer controversial. Jewish men migrating into Europe formed communities that incorporated local women, women who converted, married, and became foundational ancestors. Over generations, these maternal lines multiplied. Not evenly, not randomly. A few women left an outsized genetic legacy. They became, quite literally, genetic matriarchs. Language is a genetic metaphor. This dual ancestry found an uncanny parallel, not in biology, but in language. Yiddish. At first glance, Yiddish sounds Germanic. But listen closely. and another structure emerges. German grammar, Hebrew religious vocabulary, Slavic influences layered and later. A linguistic hybrid born of migration, adaptation, and memory. Yiddish did not replace Hebrew, it coexisted with it. Just as European maternal DNA did not erase Near Eastern paternal ancestry. The language mirrored the genome. Not pure, not chaotic, structured hybridity, isolation without stagnation. Despite living among Europeans, Ashkenazi communities remained socially and religious distinct. Endogamy, marriage within the community, was common, often enforced by law or necessity. This limited gene flow, but it also preserved internal structure. The result was not genetic isolation in the absolute sense, but relative isolation. Enough contact to integrate, enough separation to remain distinct. This balance amplified the founder effect. Traits present in the founding population became statistically prominent, including disease-related mutations, the tragedy written in genes. By the 20th century, physicians began noticing patterns. Certain genetic disorders appeared disproportionately among Ashkenazi Jews. Tay-Sachs disease, Gaucher disease, familial dysautonomia, BRCA1 and BRCA2 mutations. At first, this was framed as vulnerability, but genetics reframed it as history. These conditions were not caused by Jewish genes. They were the consequence of demographic math. Small population, rapid growth, limited early diversity. Mutations that might remain rare elsewhere became concentrated here. A tragic outcome, but also a scientific opportunity. From suffering to medical insight. Because these mutations were easier to track, Ashkenazi populations became central to genetic research. Carrier screening programs emerged. Preventive medicine advanced. Cancer risk assessment transformed. What began as historical accident became medical progress. Once again, DNA preserved what history obscured, breaking the myth of purity. Perhaps the most important consequence of this research was philosophical. It dismantled the idea of genetic purity. Ashkenazi Jews were not a sealed lineage. They were a living record of adaptation. Men carrying Near Eastern ancestry, women carrying European ancestry, culture holding it together. Identity did not depend on genetic uniformity. It depended on continuity of memory. Why this case changed everything. Before the Ashkenazi data, genetic ancestry was often imagined as linear. One people, one origin, one path. The Ashkenazi case destroyed that illusion. It showed how identity could persist despite, and because of mixing, how survival required flexibility, how history shaped biology, and biology reflected history. But this insight raised a deeper question. If identity could survive genetic transformation here, what about communities even further from the original homeland? In the final block, we move beyond Europe, to places where DNA and identity diverge even more sharply. Beyond blood, when DNA and identity diverge. If the Ashkenazi story complicates the idea of ancestry, other Jewish communities challenge it entirely, because in some cases, genetics and identity do not align at all. And that forces a final, uncomfortable question. What actually makes a people to people? The communities that don't fit the model. As geneticists expanded their research beyond Europe and the Middle East, they encountered populations that resisted easy classification. Beta Israel, the Jewish communities of Ethiopia, Jewish groups in India, such as the Bene Israel, the Kai-Feng Jews of China. Historically, these communities preserved Jewish law, ritual, and memory, often in isolation from the broader Jewish world. But genetically, many of them showed limited connection to Levantine ancestry. For traditional models, this was a problem. If Jewish identity was genetic, these groups shouldn't qualify. If it was purely cultural, then DNA shouldn't matter at all. Genetics refused to pick a side. Beta Israel, identity without genetic continuity. The case of Beta Israel was particularly revealing. For centuries, Ethiopian Jews practiced biblical laws, observed festivals, and maintained oral histories linking them to ancient Israel. Yet genetic studies showed that their ancestry largely matched local East African populations. Only faint traces, if any, of Middle Eastern lineage appeared. At first, critics argued this disproved their Jewishness. But genetics told a different story. It showed that identity can survive without biological continuity, that tradition, law, and collective memory can outlast genes. And when Beta Israel communities migrated to Israel in the late 20th century, modern DNA testing did not erase their identity. It reframed it. The Chinese and Indian Jewish experience. Similar patterns appeared elsewhere. In India, Jewish communities exhibited strong genetic overlap with surrounding populations, while preserving distinct religious customs. In China, the Kaifeng Jews lost much of their ritual practice over time, but retained family memory of origin. In both cases, genetics recorded integration. Culture recorded continuity. Neither alone told the full story. The final synthesis, a mosaic, not a line. By this point, the genetic evidence had done something remarkable. It had not proven a single origin story. It had destroyed the idea that such a story should exist. Jewish ancestry was not a straight line from antiquity to the present. It was a mosaic. Fragments of Levantine DNA, European maternal lines, African, Asian, and local contributions, bound together by shared history, exile, law, and return. The genetic core was real, but incomplete. It explained origin, not identity. Why this discovery changed science? This research didn't just transform Jewish history. It reshaped how scientists think about human populations. It showed that genetics can trace origin, but not belonging. Culture can preserve identity without biology. Migration reshapes DNA, but not necessarily memory. The old models, race, purity, fixed lineage, collapsed. In their place emerged something more accurate. Human history as a process of survival. The before and after. Before this genetic breakthrough, identity was often framed as inherited. After it, identity became understood as constructed, through shared experience, trauma, and continuity. DNA could tell us where people came from. It could not tell us who they are. That distinction changed everything. The final reflection. So what does this mean for us? It means that history is not written only in books. It is written in chromosomes, but also in rituals, language, and memory. It means that ancestry is not destiny, and belonging is not a blood test. The Jewish story, revealed through genetics, becomes a mirror, a reminder that human identity is resilient, that people survive not by remaining unchanged, but by adapting without forgetting. And perhaps the most unsettling discovery of all is this. The deeper we look into our DNA, the less simple our origins become, and the more human we appear.